students, we can conclude by saying that if there is a presence of reducing sugar into your sample solution, after addition of Benedict solution, your mixture will change its color from blue, that is the color of Benedict solution, to reddy brown, that is copper oxide, and then finally it can be changed into brick ready precipitate. You're going to see the precipitate on the downside of your beaker, or into any place that you have already placed that your mixture or your sample. You're going to see this precipitate. So ukiona ready precipitate, ujue kabisa hapo hiyo ni reduced copper 2 sulfate, copper oxide, ambayo sasa inaonyesha kwamba kwenye hiyo mixture yako au kwenye hiyo sample yako there is a presence of reducing sugar. So now let's proceed looking on another thing, that is biochemical test for non-reducing sugar. So now let's see this biochemical test for non-reducing sugar. Je, hii biochemical test for non-reducing sugar ipoje? Tunasema kwamba biochemical test for non-reducing sugar, a non-reducing sugar is a carbohydrate that is not oxidized by a weak oxidizing agent in a basic aqueous solution. Kwa hile carbohydrate ambayo inakuwa haina uwezo, it is not oxidized by a weak oxidizing agent in a basic aqueous solution. Kwa sababu gani haiwezi be oxidized? Kwa sababu tu ya lack of the presence of aldehyde group. Kwa hiyo sasa ndio ambayo tunaita sasa ni non-reducing sugar. So this non-reducing sugar in the basic aqueous medium, non-reducing sugar do not generate any compounds containing an aldehyde group. Due to that, it cannot donate electron to other molecules, hence cannot act as a reducing agent. Kwa hiyo sasa hizi non-reducing sugar, my student, keep it into your mind. Examples of non-reducing sugar is a sucrose. So ile sucrose tunasema ni non-reducing sugar kwa sababu gani? Did not generate any compound containing aldehyde group in its structure. So due to that, it cannot donate electron to the molecule and hence it cannot act as a reducing agent. Chemical or something, in order to act as a reducing agent, it must donate a pair of electrons to other molecule. So due to lack of that ability which is possessed by this sucrose, we can term it as the non-reducing sugar, since it lacks all of these properties. It lacks all of these mentioned properties. So if you will see that, then you will notify that there is absence, or there is absence of the presence of, there is presence of non-reducing sugar. So sucrose is the most common non-reducing sugar. The linkage between glucose and the fructose unity in sucrose, which involves aldehyde and ketone group, is responsible for inability of sucrose to act as a reducing sugar. This is a very simple answer. If you have been asked that why the sucrose is non-reducing sugar, while other disaccharide they are reducing sugar, the answer is the sucrose is non-reducing sugar is just because the aldehyde group and the ketones group, which they are found within their structure, they are used in the formation of glycosidic bond. And by doing that, we can say that it is responsible for inability of a sucrose to be non-reducing sugar. Haina tena uwezo wa kudonate a pair of electron to a surrounding environment. So due to lack that ability, niyo ambapo sasa tunaita hii non-reducing sugar, hii sucrose ni non-reducing sugar. kumbuka vitu ambavyo vinatengeneza sucrose kwanza, ambayo tumesema kitu cha kwanza hapa kinaitwa glucose, kitu cha pili pale kinaitwa fructose. Kwa the combination between this glucose and the fructose, ambapo sasa zile aldehyde group na ketone group ndizo ambazo zinatumika kwa upana zaidi ili kutengeneza ile bond, so that ndipo ambapo sasa inapelekea non-reducing nature of a sucrose. I hope now it is clear. And a non-reducing sugar does not reduce copper 2 sulfate. Therefore, there is no direct test for it. Kwa hiyo sasa hatuwezi tukatumia direct non-reducing sugar, that is copper 2 sulfate. Kwa sababu gani hatuwezi tukatumia direct copper 2 sulfate? Yani tukachukua tu copper 2 sulfate then tukaadi kwenye ile mixture yetu au kwenye sample yetu then tukaobserve. Kwa sababu ile tunasema kwamba non-reducing sugar it does not reduce copper 2 sulfate. Haina uwezo wa kutumia au wa kureduce ile copper 2 sulfate kuja kupata direct copper 1 sulfate. Kwa hivyo basi tunasema kwamba if it is first hydrolyzed to its constituent monosaccharide, it will then give positive Benedict test result. Kwa hiyo sasa ndipo ambapo tunatakiwa tu hydrolyze to its monomer, ambazo monomers monosaccharides of the sucrose, hapa tunaongelea glucose pamoja na fructose. So tukizipata zile glucose na fructose ndizo ambazo sasa zitaenda kuletea positive result za Benedict test. So how can we hydrolyze? We can say that the basis of test of a non-reducing sugar can be hydrolyzed by heating the dilute hydrochloric acid to give glucose and fructose, both of which are reducing sugar. Kwa hiyo sasa hapa sasa kwa kuwa hatuwezi kutumia tu direct ile Benedict solution ambayo ndio copper 2 sulfate, kwanza tunachotakiwa identify if there is a presence of non-reducing sugar. Kitu cha kwanza hapa tumesema kwamba unachukua ile mixture yako, ile sample yako, you are going to heat. Baada ya kuheat, thereby ndipo ambapo utachukua dilute hydrochloric acid uta add into your mixture. Ukisha add into your mixture, sasa zile components zilizotengeneza ile sucrose zitavunjika kuja kupata glucose pamoja na fructose, ambazo sasa zile glucose na fructose ndizo ambazo tunaziita reducing sugar. Sasa hapo ndipo utakapoenda kutumia ile ambayo tunaita sasa the Benedict solution kutest if there is a presence of starch or not. If there is a presence of reducing sugar or not. So now here you can see that the solution in neutralized with a dilute sodium hydroxide or potassium hydroxide so as to give the reducing sugar result with the Benedict test on heat. So now baada sasa ya kuadd ile hydrochloric acid, hautaenda kuiadd ile Benedict solution direct, kwa sababu umesha add ile dilute hydrochloric acid, ni acid. Kwa hiyo sasa utatumia another chemical ambayo itaenda kuneutralize ile acid, na hiyo chemical ambayo itaenda kuneutralize ile acid ambayo tutatumia hapa ni sodium hydroxide. Kama haitakuwepo sodium hydroxide, utatumia potassium hydroxide. So the use of these two alkaline or these two bases in order to neutralize the acidic from the hydrochloric or that dilute hydrochloric acid. So now you can see as to give the reducing sugar result with the Benedict test on heat. Kwa baadaye ukiheat ndipo ambapo itakuletea majibu sahihi, kwa sababu tu umebadilisha hiyo ambayo tumesema. So now my student, you can see there is importance of knowing all of these before doing the practical. So ukielewa huku, kule tu unaenda unafanya practical yako, kwa sababu everything you have, majibu yote unayo kichwani. Kwa ukienda kwenye practical inakuwa ni very simple. Unaenda tu una test if there is a presence, unasema tu there is a presence. majibu yote unayo kichwani. Why? Kwa sababu hiki na hiki. So that's all about on how we can test the presence of reducing, non-reducing sugar and reducing sugar. And by saying so, my student, this marks the end of our today's session, and keep watching our media so that you can benefit a lot of theories concerning about the food taste. And thank you for following us at Educare TZ. Bye bye. Have a nice moment.